this is part of what the GCA convention would normally be. However, we are under COVID impact. We're having to make some changes and be able to present our educational opportunities on the fly for our members. And this is a project that we wanted to get started. This will be a series of webinars that we'll conduct each month for the next several months. We've got some great programming coming up, but all in all, tonight we are very fortunate to have folks from Neogen Corporation who are able to join us tonight. In fact, we'll be hearing a presentation from Mr. Clint McWaters, and that is McWaters. Hopefully my Texas accent didn't cut it off again like it did earlier today, but again, Mr. Clint will provide that update. But before we get into that tonight, we can't bring these types of educational programming on to our members and make this an opportunity for them without sponsors. And tonight we're very fortunate to have the sponsorship of Ms. Kendall Watley. And at this time, I'd like to allow her to speak and give a presentation on what her new venture is and what services that she can provide for your operation.
Awesome. Thank you so much. Am I unmuted or do I need to do anything?
You're good. You're good.
Good to go. Okay, good deal. Well, hello, everyone. Like he said, I have the blessing of sponsoring tonight's webinar, and the topic tonight, genetic testing for commercial cattle herds, is near and dear to my heart, and it fits in with my services perfectly. The first of those services that I'm going to talk about is genomic testing consultations for purebred and commercial herds. I feel like in the commercial sector, it's so vastly underutilized and almost irreplaceable as a tool to add to your arsenal for breeding decisions, marketing decisions, culling decisions, and I'm sure Clint will get into all of that. But it would be an honor of mine to be able to help you to get started on the genomic testing journey, or add to what they're doing at Neogen, just to help you get started and figure out kind of what that means for your herd and how you can use that information. The next of the services that I want to talk to you about is photography. I do sale photography and also farm and ranch lifestyle type photography. You can see samples of my work on my website or any of my social media, and I'll give you guys the contact information for all of that in a minute. But I just have a passion for telling your story as an individual producer, whether that is helping you market your animals with sale photography, or if that's just coming and capturing your livelihood on your operation, more in the day in the lifestyle type photos, just for your family to have for years to come, to be able to remember what you did, 50, 60, 70 years ago, because if we're not taking those photos today, we won't have them to look at later. So the next thing I wanted to talk to you about that kind of goes hand in hand with the photography is ad design. So whether you're trying to promote an individual animal, or if you're trying to sell a group of cattle, I would love to help you put together an ad that best fits your services that you can use to hopefully help you get a little bit more money on those cattle. And all of the aforementioned services, whether that's genomic consultations, the photography, the ad design, I don't have any geographical barrier on any of those. I can call you, I can come to you wherever you're at. So the last one I wanted to talk to you about was that I'm an independent rep with ABS. So I sell semen, breed cows, and in Southwest Georgia and Central West Georgia, I fill tanks. So if you've got a semen tank you need filled with liquid nitrogen, I'd love for you to give me a call. So I'm just going to give you a few options for contacting me. I don't want to keep you long because I know the content tonight is going to be really great. So like I said, my name is Kendall Singleton Watley. I own and operate Bar K Cattle Services. You can add me on Facebook. My name is just Kendall Singleton Watley on Facebook, or you can like my page on Facebook, which is just Bar K Cattle Services. I also have a website, which is www.barkcattleservicesllc.com. or you can follow me on Instagram, which is kendallsw.bar.k is my Instagram handle. So, and my cell phone number is 706-741-0628. If I can help you in any way, I would love for you to holler at me. So tonight, that's all I've got for you. I know the content's going to be great, so I'd love for us to get into it.
Thank you, Ms. Kendall. For those of you, we also have some other people joining as we go, and we'll be allowing them in. We are going to put everyone on mute throughout tonight's event, but if you do have a question, we ask that you add those to the chat box or submit those through email to dale@gabeef.org, or at the end of the presentation, when we open up the question and answer session, we'll open up the microphones and allow others to speak. Again, tonight, as part of tonight's event, this is also going to serve as part of the NCBA Cattlemen's Education Series. At the end of tonight's presentation, beginning tomorrow, we'll be sending out a survey for you to complete, just to give us some feedback on how this presentation went tonight, also what you would like to see in the future. So we ask that you please take a moment, conduct that survey. It'll be online, so we'll ask that you go ahead and complete that for us. But we are providing tonight's opportunity. One of our other sponsors is the NCBA Cattlemen's Education Series. So with no further ado, let's get into this evening's let's get into this evening's opportunity here. Tonight's presentation is a thought-provoking look at how genetic testing could help your cow herd. We focus a lot on EPDs of our bulls, but we often overlook what we're matching that bull up with at home. And so tonight, Georgia Cattlemen's Association is pleased to present to you Mr. Clint McWaters from the Neogen Corporation. At this time, I'll call on Mr. Clint and allow him to take it away.
Well, thanks, Dale, and we certainly appreciate the opportunity from Neogen and in our behalf to hopefully give you guys some education on the genomics and the profiling that we have in the industry today. That's really pretty new to the industry. I certainly want to give a shout out to Ms. Kendall for her sponsorship. I've had the opportunity to meet and get to know her over the last couple of years here. If you guys need any services done, she's certainly a hard-working, knowledgeable, honest lady that I think could certainly help a lot of operations out. So we certainly appreciate it. We thank the Georgia Cattlemen's Association. Unfortunately, we have to do it this way instead of having it at the convention, but hey, we adapt and overcome and try to do the best job we can. So we hope we can give you an educational presentation tonight about genomics, maybe give you a few things to think about, and maybe refresh a few things that some of you already know. And so that's kind of our purpose here tonight. So let me see if I can share this screen, and we'll kind of start into the slides, and I can introduce our guests that I've got on with me here in just a minute. Good, Dale? So as Dale said, I'm here representing Neogen. That's who I work for. We have a partnership here, partnership to reduce risk in the cattle industry, and that's something certainly that we're going to get more into and what we hope to focus on here. Just talking a little bit about our company, kind of quick from the 30,000 foot level. We kind of operate, as we like to say, from the gate to the plate. Neogen has kind of three different departments. We've got a genomics department headquartered in Lincoln, Nebraska. That's through all of our genomics. So we not only do beef cattle, but about 40 other species run through that department. We've also got places worldwide, and we've got some labs worldwide, but ultimately all the genomics come back to Lincoln, Nebraska. We've got the animal safety division, where we get into the veterinarian instruments and cleaners and disinfectants. We also have some water quality treatments and operations through that, and that's headquartered out of Lexington, Kentucky. And then our main corporate office and our food safety headquarters out of Lansing do a lot of different testing in terms of pathogen testing in packing houses or retail sectors. And so we kind of spread across from start to finish through the whole entire beef cattle industry. Just a quick update slide about myself. I was raised in South Florida, down in Okeechobee. Graduated Oklahoma State in 2000. Was part of the judging teams at Clarendon College in Oklahoma State. And I lived out west for about 16 years, primarily working for consolidated beef producers, where I marketed fed cattle on behalf of the feed yards. So it was in that game week in and week out. Started with Neogen in June of 2019 and covered the Southeast Territory, as you can see there, with about 5 different states, and live in Sebring, Florida with my wife and daughter. And next, I'm going to introduce Mr. Kenny Stalker, and I'll let him kind of speak about himself, and then he can turn it over to Ms. Quarter.
Thank you, Clint. It's a pleasure to be here tonight. So I come from a small town in western Nebraska. So I was born, raised in a little town of, I think there's less than 100 people there today, but you know, that's gave me an opportunity to really get ingrained into the beef production part of the world, and that's what I've been fortunate enough to spend my entire career, from the feedlot sector to the commercial purebred operation, all the way down to marketing. Spent a few years on the marketing side of high-end genetics. So, you know, I joined the Neogen team back in April of 2018, and I'm got a very great position that I get to travel the United States and assist with some of the greatest team in the genomic space. We have the largest genomic team, and I get to go around and help and take care and assist where needed with that. We have 11 different territories right now that we're able to help producers, from sampling to going over their data, and just really offer a customer service piece that's second to none in the industry. So I've been doing that since April of 2018. Lovely wife of 22 years that really just helps me keep in line, takes care of the family while I'm on the road, and 2 daughters. Jackie, who's 21, going to the University of Nebraska, getting an ag business degree, and my second daughter, Delaney, she is a junior in high school, and she hopes to be a nurse someday. So I look forward to this time we have together, and I think we'll, from the presenters to the attendees, well, we'll all learn something, and I'm excited to be here. So thank you, Clint. I'll turn it over to Jamie.
Thanks for that, Kenny. So my name is Jamie Quarter. I am originally from the small town of Henderson, North Carolina. They tell me that over the years my accent's kind of gone away, but I'm hoping that if I'm at least virtually able to be with some people from my neck of the woods, that may come back a little bit as we go through this presentation. I did get my undergraduate degree from NC State, but decided that I wanted to continue my education to learn more about beef cattle genetics. And there's really no better place to do that than the Midwest. And so I did receive graduate degrees from South Dakota State and the University of Nebraska. I'm spending quite a bit of time at the U.S. Meat Animal Research Center here in Nebraska. I liked it so much that I decided that I wanted to stay. So following my graduation with my PhD in beef cattle genetics, I started at Neogen in July of 2018. And I am the beef product manager. So what that means is I'm in charge of new product development and marketing for our beef cattle products. So be that the commercial or the seed stock industry, I'm very heavily involved in that, and bringing that technology and educating our customers on the importance of it within their operations. I currently reside in Valparaiso, Nebraska, with my husband. I can't compete with Kenny of 22 years. I think I'm more at like 8 months, but I'm hoping that we'll be able to say that one day. We do spend most of our spare time in Stewart, Nebraska with our own cattle, and that's where we like to spend our time when we're not doing our adult jobs, I guess I should say. So Clint has tasked me with the fun job of kind of getting down in the nitty gritty with some of our basic genetics terminology. Although we like to talk about cows and the impact of the what we offer to you guys, it kind of goes without saying that you need a basic understanding of how the technology works. And so if we think back to high school biology, you know, basically the DNA is made up of 4 simple letters, so to speak, and we call those letters nucleotides, A, T, G, and C. And it's the combination of those nucleotides that make up genes. So the definition of a gene is a basic unit of inheritance. And that can be a single nucleotide that codes for something like coat color, or it can be a long sequence of nucleotides put together that are related to things like feed efficiency or low birth weights and things that we like to select for. You'll hear us talk about something called an allele. And that is just kind of how we refer to a specific form of a gene. So there's an allele for polled cattle and there's an allele for horned cattle. And, you know, along those lines, we talk about alleles that are identical or the same. So homozygous is when they are the same and heterozygous is when they're different. So again, I like to go back to coat color because it's just an easy one to understand. So if an animal's homozygous black, that means that they have 2 identical alleles that code for black coat color. Where we start getting red mixed in is if we have maybe a heterozygous carrier for red. So they have 1 copy of black and 1 copy of red, maybe unbeknownst to us. But that's when you have the 2 different alleles. And then finally, and less common, you'll hear people talk about a locus. And I kind of do my word patterns there. So locus just stands for location. So when we say that, that's just where that gene is located on a chromosome. But of all of those, the most important and probably most common terminology that we use today and we hear about is a SNP. And so SNP stands for single nucleotide polymorphism, not to be confused with the TSUs that we take out of cattle's ears in order to do DNA testing. And I think Clint will get more in depth on that in a little bit. But polymorphism really just stands for many forms. And so the SNP technology is what all of today's genomics is based off of. Without SNPs, we wouldn't have different genes. We wouldn't have the insight into how cattle perform that we do today. So as an example, here it just looks like a bunch of letters, right? So each one of those, like I said before, would be a nucleotide, but they're different combinations of nucleotides that code for a gene. And what just got circled, you can see here that the one difference between gene 1 and gene 2 is that there's a T that became a C. And so that's that SNP difference that then triggers geneticists who are either developing tests or are putting them into genetic evaluations that help us decide what version of a gene or which allele that cow inherited. So again, to go back to my coat color example, this animal looks to be heterozygous for this specific gene. And so that may be, you know, that they're a carrier for the red allele. You know, to be more complicated, it can be something to the effect of the top codes for low birth weight in cattle, while the gene on the top may call for, on the bottom, may call for high birth weight. But it is the reason that people like me have jobs and the reason that geneticists are really good at what they do is that it's not as simple as just this one allele that codes for a difference in those really complicated traits. It's hundreds of genes that work in combination with one another that result in the phenotypes that we see day in and day out. So to kind of bring that all full circle, where does this DNA come from? Well, the DNA comes from the chromosomes that are in the nucleus of every cell. And when we think about, the inheritance of genes, how they're passed from parents to offspring, each calf has one copy of each chromosome they get from their sire, and then one copy they get from their dam. And that's what unravels into these sequence of nucleotides, are those chromosomes. And so, like I said before, the inheritance of the chromosome from the sire to the offspring, and that those groups of nucleotides are what contain the genes that code for the phenotypes that we see in those animals. And then the variation are the SNPs. But how does all of this fancy wording relate to cows, right? So it takes a lot of research and understanding to be able to, at the molecular level, be able to divulge into the DNA of a cow and really understand what genes code for what, right? There's over 30 pairs of chromosomes. There's over 3 billion SNPs in a cow. And for those of you who are familiar with how the technology's progressed over the years, we've been working at this for a really long time. And while I'll say that we've come a long way, we still have quite a bit of a ways to go to fully understand the interactions of those 3 billion SNPs. But what got it all started was the famous line 1 Hereford cow known as Dominette. So for those of you who don't know about her, she came from the line 1 inbred Herefords out of Fort Keogh. She was selected because she was the most inbred Hereford in the herd. And the logic behind that was that if an animal's inbred, then they are homozygous at most of their locuses. But if you think about that, if they're homozygous, then that means they have 2 copies of the same allele. So when we look at genotyping her whole sequence, so 3 billion SNPs, it makes it a lot easier to put together when all of the pieces are the same. But that project was started in 2003, and it took over, at that time, $50 million, 300 scientists across 25 countries. But we are happy to say that the first version of her genome was published in Science Journal in April of 2009. And this was really a huge stepping stone for the beef cattle industry, and it was one of the first livestock genomes to ever be fully sequenced and understood. And so after that article was peer reviewed, her genome was published into, basically all of academia. So all of the research that's been done since 2009 has been mapped back to this process, to this genome. And this process itself is what allows for us to really do research into the SNPs that make up the chips and all of that fancy stuff that we make at Neogen that help geneticists create EPDs and know which portions of the genome code for specific traits of interest, like weaning weight, feed efficiency, and calving needs.
All right, thanks, Jamie. So now that we all have the foundation, we know what all the acronyms mean, and we have a good understanding of how the technology and how the science came to the industry. But how do we implement it? What sectors of the industry are using genomics? And as you can see on this slide here, we touch every single sector of the entire industry with genomics at some level. At conception, that's where it all starts. We have AI companies. We have our elite seed stock. They're using genomics to find the outliers. They're using genomics to find the next greatest animal to increase their genetic growth from where they are today to the next level. As we continue, our population continues to grow. As our genetics continue to grow, they have to increase, and we don't want to be left behind. So it all starts at conception. It's our AI studs finding the best sires through genomics. We're not using parental averages anymore. We're actually using the genetic testing to find the elite sires, the elite dams, to multiply the genetics to the next level. Then it moves to the commercial herd. This used to be a technology technology that only the seed stock has implemented. Well, today it's moving to the next sector, the commercial herd. How can you take those females or those steers that you're going to go to market, and how can you help the next level select the best individuals for the rail, or for the keeping the best individuals to stay at home on the ranch to continue to move your genetic progress faster? Is it selection for stayability? Is it selection for fertility? Longevity. Those are all traits that are very impactful for our commercial producers. And we have some of the largest operations in the United States: the Deserets, the King Ranches, the Parkers, the Trues. They see the value of it, and they're going to continue to drive the genomic piece on their operations because they see how much impact it has made from generation to generation. The next level is the feeders. How can we help a feedlot be more efficient in the way they manage their operation, from feed efficiency, average daily gain, to carcass merit? Those animals that are going to get a grid premium, those animals that are going to finish 30 days sooner. Every penny, every day in that yard is impacting their bottom line. So it doesn't take them to purchase a group of calves 1 year to understand what the potential is for that yard or for that group anymore. If those calves are genomic tested on arrival, they know what they're going to do, what they're going to average on gain, or as an individual, what they're going to hang on the rail. So all of a sudden, we've taken a lot of the guesswork out of the feeding industry, and we've helped them have a much more reliable inputs on what that group's going to take. Now, the distributor, the retailer, this really comes down to two sectors of Neogen. From the food safety, measuring for pathogens, to disinfectants. How can we help our food be the safest there is in the world? Going back to on the genomic side, we're helping them be able to see what their supply chain is coming behind them. They know what their demand is, what's actually in the pin that's behind them, that they have a good understanding of what their ribeye size is going to be, what their carcass and their marbling is going to be. All the way to the marketing channel. There's where are these animals? How can we add value to these feeder cattle that are going out there? And Neogen has partnered with two of the largest genetic value-added companies there are, is Top Dollar Angus and Superior Livestock. So these commercial producers, they want extra value in their operation. Well, let's prove it. If you have the best calves, the genomics are going to prove that you do. And why not? If you're taking and you're investing in that value, why not market that? So through genomics, we're able to give them value-added marketing channels that they can go and get premiums for their cattle. And maybe if they're not there yet, they'll have a good understanding of where their cattle are, where they can go back to their seed stock supplier and say, I'm missing this piece. How can you help me? And that's where the seed stock guy comes into this chain, is their ultimate goal is the commercial cattleman. So once they've already genomically tested their animals, they're able to market and sell very high accuracy bulls into their commercial customers, but they're also able to take it and dive in one step closer into the whole genetic growth. Is they're able to look at their commercial customers and see where they can do to enhance their operation to the next level, all the way through the packer. That's even more going down the value added branded beef from NHTC, from those channels, from the more breed specific. All those can be measured through genomics to help them on the distributor side, as well as the chain coming up behind going into the packer, and then all the way down the consumer. Where did their food come from? There's no better traceability than DNA. You can't lose that traceability. And that takes one more sector of where Neogen is at, and that's blockchain. Through DNA, we can help the consumer know exactly what operation that animal was born at, all the way to the grocery store, to the packer. So the consumer, the value that DNA brings to the consumer is endless, because you will not lose that chain of identity there. So what options are there? As Clint mentioned earlier, this really started in the poultry, the porcine industries. It moved into the dairy, the seed stock, but it didn't stop there. It is continuing to go down the chain into the commercial cattlemen, into the feeding sector. So what products are there? Igenity Beef, that's our number one best-selling, fastest-growing product that Neogen offers, and that's for our commercial cattlemen. It's a crossbred genomic test, so it doesn't matter as long as you're one of the 8 breeds that make up Igenity Beef. This works very well to dive in to see where your genetics are on your operation. It's not just, yes, this female looks good. We can actually differentiate between 2 females that look really good to who's going to be profitable and who's going to cost your operation moving forward in the lifetime as she is in your pasture. To our GGP, that is our gene-seq genomic profiler. So this test used to be a 50K. It's going to be very soon, it's going to be 100K, and it's offered to all of our breed associations. Currently today, we work with 32 different breed associations across the United States, helping those producers to be able to select and to take their genetics from where they are today to the next level. And when you get 100,000 markers, it really helps the evaluation team, whoever's running that evaluation, really be able to differentiate and find those elite, who's the next bull, who's the next dam that's going to move that operation in the right level. And it also gives a lot of accuracy to the commercial producer that's buying those bulls. Increased accuracy on other traits. It guarantees parentage, so you're never in doubt that you are actually buying what you're buying when you go to that commercial or to that seed stock sale. And also on the same line there is our Angus GS. This is the industry's first breed-specific genomic test that was 50,000 K, but we're going to continue to advance the technology in the industry. And this last fall we updated it, and it's 75K. So this is a breed-specific test that's strictly marketed through the Angus Association to help our Angus breeders stay advanced and stay on the cutting edge with their genomics, all the way to the custom panels and arrays. As Clint mentioned, I think we currently today, we have 42 different species that we run genomic tests for, it's Neogen has always been kind of their forte, is how can we help the researchers and the developers find the chip, the panel that's going to help those specific species. So areas that DNA testing is currently used today, at trait specific, as Jamie talked about, it can be something as simple as coat color or horn pole, or to the recessive lethals that almost every breed association out there that they help their producers breed against, to try to eliminate those recessive lethals that will sometimes are very impactful on the operation's bottom line. To as simple as parentage analysis. How impactful is that to a commercial operation if you are using multi-sires in your breeding group, to be able to understand which sire out there is actually getting the job done, which sire is out there laying under a tree, all the way back to the breed associations to help keep those breed lines pure, to really understand parentage. Genetic evaluations, as we talked about, GE-EPDs, those breed associations and those breeds that are really focused on moving the needle is, taking a EPD, putting it on steroids pretty much and moving it to a GE-EPD and looking how fast that will move a breed and how fast that will move a herd when all of a sudden you are taking accuracies from 5% on a yearling bull all the way up to 50%. That is offering value not only to the breed, but to that commercial customer. And that's where GE-EPDs come in. To our genomic profiling, I mentioned our Igenity Beef. So for those commercial producers out there that they're really wanting to move the needle, but they don't have all the phenotypes that they need, or they don't have the numbers of animals that they need to turn in to get an EPD. We're able to do that on looking at a single DNA sample, looking at their molecular breeding values with very high accuracy. We can help a commercial producer have very similar predictions on each trait like a seed stock, but yet at a different level. So the commercial profile is helping move the needle on the commercial operation, and Igenity Beef is the first and the best one out there for that.
Well, thanks, Kenny and Jamie, and I appreciate the opportunity for you guys to be on and kind of go through some of the upstarts and some of the terminology as we move through here, and to kind of get into the meat of the program and what we're talking about and kind of where does this fit in the industry. I've used this slide before in several different presentations, and I think as we go to bull sales and we look at bulls and we decide what are we going to buy, and I think the standard answer is, well, I'm going to try to buy the bull that maybe has the lowest birth weight EPD or the highest marbling EPD. And I'm going to try to buy the one that I can pay that fits my budget, closest to these certain traits that I may be looking for. So the question is, the most expensive bull always better, and does a cheaper bull always save you money? We're going to come back and try to kind of answer that as we go through this presentation and give you some food for thought as we come through there. I think to begin with, and we're talking commercial cattlemen here tonight, but in order for the commercial cattlemen to know and understand what the seed stock guys are doing, I think we need to go over it real quick and give you a better understanding when you go to buy bulls at these seed stock operations. The main thing that the seed stock guys do it for is they have a lot of different things, as you can see here, but the very bottom one, improve customer confidence. And that's really and truly what they're getting at and what they spend the time and effort and money for. Yeah, they want to make their own cattle better and they want to manage genetic conditions. But at the end of the day, they want to improve your confidence as a commercial bull buyer. Use of genomics in terms of seed stock and in terms of registered operations. Previous to genomic profiling, we used EPDs, and they were made up of 3 different things: pedigree, phenotype, as you see on the left, and then progeny information that comes in from that animal that you see on the top right. When we started out in genomics into the industry and were able to start testing animals and determining what genes they actually inherited, then we were quickly able to increase the accuracy of these animals and have a little better idea of what they're going to do. And an example of that that's really good that I like to use is a set of ET flush mates would all have the same EPDs until they put some of their own individual performance in there. Well, a set of flush mates, ET calves at a day old or 2 days old, we can run a genomic profile and actually figure out which ones maybe got the low birth weight genes, which ones got the high growth genes, the high marbling genes, so on and so forth. And that becomes very usable. This is kind of an example of what we're talking about when a seed stock guy or a registered operation uses a genomically enhanced EPDs. Is the bottom here is the accuracy of when you're buying bulls, and that 5% would be on a virgin bull or a yearling bull, a 2-year-old that we typically buy. If we run a genomically and genomic profile, combine that with their EPDs in the background, we can increase that accuracy depending on the trait and the heritability. But in general, we can increase that about 20%. That makes 25% accurate for a certain trait. And once again, we're talking about virgin bulls here with no progeny data on track. Consequently, if we kind of run a middle-aged bull that may be up in AI stud sire somewhere that has some progeny, has some information on him, he may be at 50% accuracy. And if we run a genomic profile on him, we may only add 5% to that accuracy of that particular trait. If we get a bull that's been around for quite a while and has a lot of progeny information, a lot of data on file, we've got a lot of information on him, we add and pull a genomic profile, we're not going to add anything to his accuracy. Why is that? We already have the information. It just took us a long time to get there, but we already have it. The important one for us in the commercial industry is this one over here, because we can buy bulls that have never sired an animal or heifers that have never had a calf, and we can actually increase our chances of getting the trait that we are looking for by increasing this accuracy prior to them ever having progeny on the ground. And a lot of people look at that and say, well, that's only 25% accurate. That means he's still 75% wrong. And that is some truth to that. But what it does is it narrows the range of how far wrong is he from the printed EPD that we have and the GE-EPD we have on his papers. And usually what I would say is if I'm going to play the lottery and have a chance to win a couple $1 million, do I want a 5% chance or do I want a 25% chance? At the end of the day, I'll take my chances with 25 versus 5. And that's kind of the way we look at it as we run through this. Another side on the seed stock side, just to show the variability that an animal can have, is we always think about, well, if I just breed this bull, he's a good one. He's going to have a bunch of good calves. And there's some truth to that. But at the same time, there's a lot of variability in this genetic makeup. And so as we see this example for the trait CEM, Cavanese maternal, all of these are sired by the same Angus bull. And these are embryos that we took genomics from. And we can see that one bull here is in the top 10% for the breed, and one bull is, in the bottom 98% for the breed. So even with the same sired animals, what we do with genomics is go and find those bulls, our cows, our heifers that have the genetics that we want, and we're lucky enough to get what we're looking for. And there's a lot of variation even within siblings on this. What can we not see? I think it's super important as we learn and we educate ourselves about genomics to understand that genetics plus environment equals phenotype. And the phenotype is another term that you'll hear about in genomics and genetics. And phenotype, as I was taught back in the day, is how good does an animal look? Well, as we talk about it in genetics and genomics, phenotypes are measurable traits, i.e. weaning weight, yearling weight, birth weight, measurable ribeye sizes. So phenotype, you have to have the genetics and you have to have the environment to express those genes to create your phenotype. And that reduces risk and it increases efficiency for you. And a good example of that is if we're in South Georgia and we're on that good grass and we've got a cow that has a calf and it's the same genomics in that animal, will that animal weigh the same as if it's in my good friend Tyler Gray's area in the desert of Nevada? That's probably going to be two different weaning weights on a calf that has the same exact genomics, because environment's going to play a role in that. And we've got to remember that and understand that. The Igenity Beef profile, it says new here, and it's not that new. What is new is we've partnered with IGS, which is greatly going to increase the amount of different genomics, genotypes and phenotypes that we can kind of pull across that panel. And in the future, hopefully increase our accuracy and increase our correlation with our tests by having all that information with IGS's partnership. Igenity Beef really helps you rank and select replacement heifers, and that's really what it's built for and the goal. But there is some additional uses to it where we can use those genomic profiles in a cattle herd to produce certain feeder cattle that fit your market. He mentioned Top Dollar Angus and Superior. Those are marketing programs that we look at the about 35% of the calf crop, and allows us to start getting an idea of what are the genomics in that calf crop and in that herd. And when we understand what we have within our cow herd, then our buyers are going to be more willing to pay more money. Once again, why? Because we reduce the risk for them and we increase their efficiency. Anytime we reduce risk in this business, we're either making money or somebody's giving us more for it. And that's usually what we're looking for. We can also use it to score some commercial bulls. And probably a huge one that is really used in the industry and probably in some cases underused is sire parentage or dam parentage. And so if we kick out that AI bull, or we AI breed, and then we kick out a bull right behind that AI breeding, we always have those calves that fall in between, and we're not real sure which one they belong to. And so we usually just kind of put down something. Now we can go in and actually make sure that when we set down an animal for a sire or dam, that we're 100% confident. The traits that are reported, maternal traits, as you see there to the left, growth traits, and then carcass traits. One thing to note in the commercial, for our test in the commercial industry, those traits are MBVs, molecular breeding value. So on the commercial side, we take in no account of what the pedigree of that animal is or what the performance of that animal's parents were, like we do in EPDs. And that's the idea behind it, is we may not know who the parents are. We may not know the lineage of that animal. And so we only look at the genes that that animal has inherited, and that's what we use. Most of these traits are used and gathered by several different companies out there and reported in different ways. One of the traits I think super important and kind of dear to my heart is the tenderness trait you see in the carcass side, because I have spent so much time in that feed yard setting and industry, and within that, I think that is going to be a trait that's really going to have some economic relevance, in my personal opinion, as we move down the line here to help our consumer confidence. And I think that's something that's a cheap trait to get that we used to not be able to get without a lot of expense. We do use identity indexes. You can custom make your own index for your operation. You have a dashboard with all of your information coming through there. You can sort any which way you want. If you got an index that fits your environment better than one we've pre-built for you, by all means, you certainly can do that. But we like to use indexes so that we don't single trait and get ourselves in trouble there. So we try to use the multi-trait index and selections for our operations. What are the different ways that you can submit DNA? And all companies out there that do this genetic profiling and genetic testing will use these type of things. The preferred method, I think, across the board is the tissue sampling unit. You heard Jamie refer to it earlier, so a TSU. That's the picture of it in the top right of one that's been collected. You can see right in front of that little green ball, there is a piece of ear tissue in there. And so that's what the lab would actually use, and there's a barcode on it. And it's about just like giving an ear tag to an animal. We can collect that. We also do blade cards, and a lot of people do those, and those are very reliable as well. And maybe a little bit cheaper in cost, but a little bit tougher to deal with in some other avenues in terms of contamination. Hair cards at the bottom there, that's kind of the old tried and true, and what we started with was hair samples. That's one maybe we're fading away from just because of the complexity of and the fail rate on that. And then we can use semen straws to pull DNA. Hair cards, they do charge at our lab $4 extra because of the time-consuming ability to create that sample. But three very good samples. The tissue sampling unit is an all-flex product. So anybody who deals in all flex, that's not a product just to Neogen, that's an industry-wide product out there for that particular case. The last thing I kind of want to talk about is a product line called Invigor. And the reason I want to bring this up is it's a way that we measure heterosis in cattle. And certainly heterosis, as we all know back to our animal science days, is super important, we believe. The things to note about heterosis is it's not heritable and will not transfer to another generation. So just because a cow has a lot of heterosis doesn't necessarily mean she's going to capture and pass along all those genes to her offspring that are the same disbursement of breed percentages. So it's not a good replacement for a breeding program in terms of crossbreeding program. The heterosis, one thing we talk about here is and I love this slide. John Jano was fortunate enough, or I was fortunate enough that he would let me borrow this slide, and it's a 931. And what we mean by that is heterosis, we believe towards the fertility side will increase the ability of that animal on the fertility and health traits stronger, and heritability gets stronger towards a carcass trait. So we can make a lot of changes with heritability and carcass traits, but we're a lot slower to get that progress done in the fertility and health traits. With heterosis, that expresses those traits much greater. And we like to say they're 9 times more important than growth traits 3 times more important than carcass traits. I've actually had some people maybe argue that with me a little bit. I guess put in any numbers you want, but I would tell you that it doesn't much matter what an animal's marbling is if I can't keep it alive to get it harvested. So I think fertility certainly makes it a pretty strong point on that end of the spectrum. A quick study on just kind of proving some heritability and heterosis, I should say. There's a 2,000 head database there that we did a program in Washington. The top 25% cattle with the top Invigor scores, meaning they have more heterosis, versus the bottom 25%. Pretty astounding numbers. Treatment for respiratory problem increased by 22% for those bottom cattle. Number of treatments increased by 28% for those bottom cattle. And when I say bottom, all I'm meaning is ones who have less amount of heterosis and crossbreeding in their program. So back to the question we started with, determining bull value. Well, the easiest way to find out which bull is the best and which one you want to buy and use is testing and knowing your commercial herd, knowing what your bell curve is. Am I really strong in growth already? Am I really strong in weaning weight or marbling already? Am I really strong in these traits? So if that's the case, maybe I need to buy a bull that's strong in some of the traits that I'm weak in. What are my economic drivers? What makes me the most money and puts the most money in my pocket? That's the traits I need to be chasing, but I don't know what to buy until I know what my commercial herd already is. And I work with a lot of seed stock producers out there, and if they do GE-EPD and genomic tests of their animals, if you will take them their genomic profile of your cow herd or your heifers year after year as you do them, they will be more than willing to help and go through. And that bull may be lot 25, maybe lot 70 is actually the best fit for your particular herd. And so that's how you make genetic progress, is knowing both sides of the equation and what you're wanting to chase. I'll kind of end it with this quote here that my friend Rick, one of my coworkers in Kansas, had a gentleman that he was working with here that said, we can now make the progress in the life of a cow that we used to take the life of a cattleman. And I think there's a lot of truth in that matter of how quick. And once again, this is a tool, guys. This isn't a be-all, mean-all, be-all, but it is a tool that can quickly help us in the commercial industry. And I agree wholeheartedly with Kendall. I think it's super underutilized, but people are certainly starting to look at it. So with that, Dale, I'll turn it back over to you on Q&A. And if anybody's still awake out there, then maybe we can have some question and answer sessions.
Thank you, Clint, for that. I apologize. We had a little technical difficulties over on my side. I couldn't get myself unmuted for once. So it happens to the best of us. Folks, remember that you can submit your questions to the chat box. You can also email them to myself. But here momentarily, we'll open up all the mics for everyone to talk. But to help get some of the more general questions out of the way, for those of you at Neogen, what's your opinion of the best time to take a sample?
Yeah, I'll take that one. I mean, it really depends on your operation, Dale. The best time, in my opinion, to take a sample typically is when you can get your hands on them. I have a lot of producers that take the sample on a day-old calf. Now, we encourage people to, let that calf be dried off and we don't have any embryonic fluid on it. But we can take it as quick as a day old. And then we know what that genetic merit is by the time that animal comes to weaning pen or at first working. Or somebody can take those animals, particularly on the heifer side, maybe sort those animals for what they would like to keep from a phenotypic looking standpoint, and then take the test if they're going to post-wean 60 or 90 days. The test takes about 30 days to get back. So just allow yourself 30 days prior to needing to make a decision.
Awesome. We've had a question from the audience, and I believe this will also kind of cover that, not just the specific test itself, but what does genomic testing cost for a commercial cattle producer, and how do you recommend that a first-time user begins to implement genomics into their operation?
Yeah, Kenny, you want to take that one?
Sure. So I'll start with the second half of that question, and it's how does a producer first look at implementing genomic testing? And that's at two different levels based on whether they're a seed stock or commercial. But I'm going to start with the seed stock. Is it important for that seed stock producer to genomically test their bulls? Absolutely, because they're offering their customers a more accurate product that they can stand behind. But it's also important to test their females. Those females are what's going to make the next generation and keep the ranch going from year after year. So it's very important for them seed stock guys to be checking their females. So they're making sure that they're selecting the best females to move forward. But then it goes to the commercial operation. How expensive is that to save this female because she really, really looks good? So we always encourage our producers, start at your heifer pen. First and foremost, select those females on phenotype because they have to work in your environment. They have to be structurally correct. They have to have the right docility. Get all the phenotypical selection done. And then if you're going to select 20%, we recommend you test 30% because we really don't care about the upper end. It's eliminating that bottom end, those bottom end females that are going to continue to be in your herd for the next 10 years that cost your operation year after year after year. So select 10, 15% more than what you want to keep and get rid of that bottom 10 to 15%. And the cost, that varies on what test you're running. From a seed stock side, if you're running Angus GS, that's $37. If you're one of the other breed associations, that ranges from $40 to $85, just based on whatever that association decides to charge their members. Down to the commercial level. For a $29 sample, you can get 16 traits, 3 indexes, parentage. You can have all the data you could ever want for $29. And that might sound a lot, but once you decide what it costs you if you lose a female in year 2 or 3, or what happens if you can bring 10 extra pounds across the scale each year. So there's a lot of ways of looking at ROI. But Igenity Beef's $29, parentage, that's $15. Clint was talking about Invigor. There's two options for Invigor. You can add it to Igenity Beef for $5, or you can order it as a standalone and it's $20. So there's lots of different ways and lots of different prices, just based on what works best for your operation and what solutions you need for your operation.
Thank you for that. You mentioned in that discussion there the phenotypical traits that we generally will select for. What are the benefits of using genomics as a tool for making selection decisions along with those phenotypical markers that we often focus on?
So the benefits is, as we said, is making your operation more efficient, looking at the bottom line. So we've all done it. We've all saved those replacement heifers that, they just look really good. And year after year, they bring in the smallest calf to the scale, or you just can't deal with her docility. So, phenotype, it's very, very important, but we call it pulling back the hide. What is her true genetic makeup that she's going to bring to the table? Feed efficiency, it costs, it's one of the highest costs of a lot of operations, is what's that feed bill going to be? If you look at feed efficiency, average daily gain, that really impacts the bottom line. And I don't know about you, but I think there's very few people out there that can tell you what the average daily gain or feed efficiency is going to be on an animal just by looking at them. And you can take it to the next level. If you're a feedlot owner and you are really, the margins are pretty tight, if you could have the luxury of buying one pin that what their average daily gain is, what their grid potential is, how much more valuable is that pin to this other pin that is never going to make a grid premium? They're going to be on the bottom end of average daily gain and efficiencies. So there's huge gains in the feeding sector. So you can't tell those measurements by just looking at them. You got to really understand what their true genomics are.
Awesome. Well, let's say I'm a producer, and after listening to tonight's presentation, I'm now firmly in I want to consider doing this. Do I need to test my entire herd, Clint, or is there a recommended amount that you prefer that we focus on?
Well, good question, Dylan. It's really up to the producer. The way we recommend doing it, and kind of the way I think most of the industry looks at it from the commercial side, is testing those potential replacement heifers every year. If I'm going to keep 20%, which is kind of a normal number, I may test my top 30% and then use that test to sort back down to my top 20% based on what the animals I would have kept just physically looking at them. If you want to and you want to get really progressive, certainly if you test the whole herd and get your bell curve in place, now we can start adding bulls that fit your herd, and we can make genetic progress much quicker. But that's not the most economic route sometimes for somebody to jump into it. So we usually start with that replacement heifer side of it. Kendall, do you have anything kind of add on that as far as the way you advise in some ways?
No, I would just concur with what you've said. Most customers, if they're wanting to keep the 20%, they'll test the top 30, 35%. And, there may be some that will go about it. As they're running females through the chute for the very first time, they may make that assessment as they're coming through the chute. I'm going to cull, the bottom 70%. And that way you've already got, those top 30% that you're going to want to test. I concur with you that if you're keeping the top 20%, it's best to just get that top 30, 35%, get data on them. That way you've got more tools in the toolbox to make those breeding and culling decisions.
All right. Well, thank you all for that. Last but not least, we're going to open up the microphone, so this may get a little interesting. but I believe that we've gotten most of the questions out there, but there may be one from the audience where somebody might be on the phone and unable to type in. So I'll unmute everyone here and then give it a couple minutes of questions, and then we'll wrap up. So if you have any other questions at this time, please feel free to go ahead and ask them.
So I have a question. Tell me the process. You said it was an all-flex tool to do, I think you said the TSU. What's the process? Do you buy that tool? And then you mentioned an Excel spreadsheet. Do you buy the tool, fill in the spreadsheet, and then mail those samples to Neogen? Or can you just go through how the process works?
Jamie?
Yeah, sure. So the answer to all of your questions is ultimately yes. So to start the process, you can either contact Clint directly, or you can contact the Neogen office and order your sampling equipment. So you just need to know how many TSUs you want to order. And then we also sell, if you saw in that photo, there was a blue gun. So just like an ear tag, you have to have the gun with the needle on it to be able to take the sample. So you would order that equipment ahead of time based on how many samples you wish to take. They come in individual boxes of 10. I would highly recommend getting those boxes because it just makes keeping everything straight a lot easier. And then whenever you're ready, you just load the gun. We have some information on how to do all that. You take samples, and each sample has an individual barcode on it. So when you download that Excel order form off of igenitybeef.com, like I had noted in the comments or in the chat box, you would put your on-farm ID in the animal ID column, so you know what animal it is, and then you put the TSU barcode number in the sample barcode column, so we know what animal that is. And then there's some other optional information that you can fill in, but basically the Excel order form, the way it works is it will basically calculate your total. And once you're ready, you hit submit, and it will email that order form to us at Neogen, and it will be awaiting for your samples to be mailed directly to us in Lincoln.
Awesome. All right, folks, we got time for one more call. We're a little over our goal here, but if anybody has one more question, we've got time for that.
Not here.
Well, I don't want to.
Go ahead.
Go ahead, Drew.
I don't want to monopolize, but if nobody else is going to ask a question. So the $29 for the basic Igenity test, would that cover? I'm sure it doesn't cover the gun, but would that cover the kit and y'all doing the sample and everything?
So the $29 price point is just the 16 traits and the 3 indexes. The price of the little TSU is basically $21 a box of 10, so $2 each roughly. And then the gun itself is $45. But I will say, the gun, they recommend to buy a new one every 500 samples. But, I always recommend to at least have 2 on hand just in case it breaks.
All right. Well, ladies and gentlemen, thank you again for joining us tonight. We greatly appreciate our representatives from Neogen, our sponsors, Barkay Cattle Services, as well as NCBA's Cattlemen's Education Series. Again, you'll be receiving a link to an online survey after the fact, after tonight's webinar. We ask that you just take a moment, please complete it. It's pretty short. It's only about 8 questions. But it'll give us a great amount of feedback that we need in order to help continue to make these webinars successful and beneficial for all parties. So with that, we want to tell each and every one of you good night, stay safe, and thanks again for joining us.
Thanks so much for having us, Dale.